Thanks, Greg. So again, my name is Surojit Paul. I'm here in the Department of Neurology at University of New Mexico, and I'm going to talk about our work on the step peptide mimetic that we have developed here in the lab. Next slide, please. So my background is I have a Ph.D. in neurosciences, and my research interest is mostly on understanding the molecular mechanisms of neurodegenerative disorders, particularly those related to oxidative stress and excitability in the brain, and then for potential development of therapeutic targets. My research has been continuously funded by the National Institute of Neurological Disorders and Stroke, the most recent funding being in 2018, which will go till 2023. It's a $2.5 million that we got from NINDS for this current research. Next slide, please. So the innovation here is a therapeutic agent to reduce brain damage, especially in stroke patients. So over a long period of time, we have been working on this group of enzymes or proteins called STEP. The interesting thing about this enzyme STEP, it's exclusively expressed in the brain. And we are always trying to figure out actually, why is it exclusively expressed in the brain? What's its physiological function? And eventually we discovered that one of the main functions of this protein STEP is to provide neuroprotection to the brain. Neuroprotection actually against the continuous stress and the trauma that the brain goes through on a daily basis. What we further realized, actually, when there is a severe insult to the brain, what happens to this protein, which gives initially neuroprotection, it degrades or it gets inactive. So if there's a huge oxidative stress or if there's an increase in excitation to the brain, this protein degrades, and so the neuroprotection is gone. So here's the protein STEP, and based on its mechanism of regulation, which we extensively studied, we were able to generate a small peptide, from this protein, which retained the neuroprotective function of STEP. And using this peptide in preclinical studies, of course, in animal models, we were able to see that this peptide, when we inject it in animals, it can arrest the progression of brain damage after, particularly in an animal model of ischemic stroke or cerebral stroke. And that's the basis for the drug discovery that we have today. Next slide, please. So what we wanted, first of all, talk a little bit about what is cerebral stroke. So cerebral stroke, primarily it happens due to an artery getting blocked, an artery which supplies blood to the brain. If it gets blocked for any reason, so what happens, a certain region of the brain don't get any more blood supply. So there's no oxygen and glucose to the brain, and that part of the brain starts dying. And so this is the type of stroke, what we call is ischemic stroke, and 87% actually of cerebral stroke are ischemic in nature. There's another kind of stroke, which is called hemorrhagic stroke, where a blood vessel in the brain all of a sudden ruptures, and this is about 10 to 15% of the stroke are hemorrhagic in nature. And hemorrhagic stroke is actually more fatal. So in case of cerebral stroke, it is the most common cause of death worldwide, first of all. Second thing, it's also the leading cause of long-term physical and mental disability. So one of the things about the incidence of stroke in individuals, it used to be that the incidence of stroke used to increase with age in the population of 65 plus. But more recently, we see that the incidence of stroke is increasing in the individual group, which is less than 55 years old. About 30 to 35% of the stroke today happens in individuals who are less than 55 years old. And a couple of reasons for this. One of the things is the lifestyle, increasing stress, hypertension, high blood pressure, our sedentary lifestyle, and the food that we take, which leads to obesity, diabetes, hyperlipidemia, hypercholesterolemia, and other metabolic disorders, which are all risk factors for stroke. So today, hypertension accounts for 54% of all strokes. And the prevalence of hypertension in American adults is around 45%. And for the population greater than 60 years, this is even higher, 72% actually have hypertension. Global hypertension actually again has increased 442 million in 1990 to 874 million in 2015, which is about a 97% increase. And the prediction today is that the incidence of stroke is going to increase by another 25% in the next 2 decades. Next slide, please. So what are the treatments that are available for cerebral stroke today? None. So there are no treatments available actually for protecting the brain against actually this insult. The only drug or the group of drug that's available is what we call activase or alteplase, which is actually a clot buster. So as I mentioned, in case of ischemic stroke, there is a blockage of an artery which is supplying blood to the brain, And so it causes the brain to start dying. So this drug activates or alteplase. What it does, it can dissolve the clot and allow the blood to go back to the brain again. So sometimes that's good, and sometimes what happens, as the blood goes back to the brain, it causes additional injury to the brain, which is called reperfusion injury. Now, not all stroke patients can get activated alteplase. It's so patients have to arrive in the clinic or in the hospital within between 3 hours to 4.5 hours after the onset of insult, which doesn't happen in most of the cases because patients don't even realize actually when they had these incidents of stroke. So the other thing is that when a patient comes even within that time period of 3 to 4.5 hours in the clinic or in the hospital, many of them have pre-existing conditions such as hypertension, such as diabetes, and they're not eligible for treatment with activase or alteplase. The reason is that activase and alteplase have several side effects, and one of them is increasing hemorrhage in the brain. So as you can see, that's the only treatment that you have, is to dissolve the clot and let the blood go back to the brain, but there's no real treatment in terms of giving protection to the brain. And so National Institute of Health is actively seeking for neuroprotective agents which can protect the brain from this ischemic insult, and then extend the therapeutic time window for treatment with alteplase, and then also can mitigate the side effects of alteplase, and then have further better functional outcome. So this peptide that we have developed, it meets all these criteria. First of all, it can be administered intravenously. Second thing is that this peptide can be dissolved in any physiological buffer, so we do not need any special buffer for this, and it can cross the blood-brain barrier when it is injected intravenously. Third thing is that this can be used along with alteplase or the clot-busting agents or strategies. It can be even used or applied actually to patients before alteplase treatment, or even in the group of patients who are not eligible for alteplase or clot-busting treatments. So it can, because it's a neuroprotective drug and it's also resistant to degradation, which means it does not get degraded in the brain. Next slide, please. So what is the potential market need for a stroke drug? So as you can see here, that currently, so from 2012 to 2030, stroke-related medical costs actually have increased. Actually, is projected to increase exponentially from 71 billion to 184 billion. And today, again, just the sales of the drugs TPA or alteplase and anticoagulants, and just for that group of drug which dissolves the clot, the market is currently in 2017 7.2 billion. Was the market in 8 major markets, which includes North America, Japan, and several countries in Europe, with 3.4 billions of drugs being sold in the US alone, which is more than 50%. So it's expected that the global stroke treatment market is predicted to be increasing. It's to be at least up to $50 billion actually by the year 2027 in the major markets. And then there is also significant opportunity to exploit the untapped markets in the emerging economies such as India and China. So there is a substantial market and demand actually for any kind of neuroprotective agent actually for stroke treatment, as there are none today. Next slide, please. So here I'm going to share with you some of the data and the results that we have. So how do you evaluate the efficacy or the neuroprotectiveness of this peptide that we have developed in today? So in preclinical studies. So basically what we do is we take animals, which are rats or mice, and we induce stroke in them. So how do you do that? So we do a small surgery in the neck area to expose one of the major arteries that is supplying blood to the brain. And then we generate a small nick in those arteries and put in a filament which will block the blood supply to the brain, to a certain region of the brain. And that region immediately gets damaged. So in this case, what we typically do, we keep that filament or block it in the artery for at least 1.5 hours, and then we remove that block so that the blood supply can go back to the brain again. In the meantime, right after stroke or even several hours after stroke, we inject this drug or the peptide that we have intravenously through the femoral vein. And then 24 hours later, we will image these animals. So these are live animals, and we image them with MRI or magnetic resonance imaging. Typically, that's what is done in clinic, actually for a human patient. And then again, 2 weeks later, we again actually image them. And then again, 28 days later, which is 4 weeks later, we image them again a third time. And then we do a series of behavioral assessment in these animals in the meantime. So here's one representative MRI scan from treated and untreated animals. And as you can see in this group here, in the top group here, top row, these were not treated with any drugs. And so what we see here, the MRI scan from the front of the brain to the back, there's an extensive damage, and this white area actually represents the damage here. And you can see there's an extensive damage here. Whereas in that group actually, which was treated with the peptide, 6 hours after the onset of the insult, there's a substantial mitigation actually of damage. And this is again represented here by this bar diagram here. You can see there's a substantial or significant decrease in infarct size. And this decrease in infarct size is maintained all the way up to a month. So that's the long-term evaluation that we have done so far. Another interesting thing that we observe here actually, that there is also substantial decrease in mortality. In the vehicle-treated animals, within the first 48 hours, out of the 16 animals that we started off with, only 10 survived and 6 died. Whereas in the peptide-treated group, only 1 animal died, and then the rest lived all the way up to 1 month. Next slide, please. So another group of animals that we have started testing is the hypertensive group. So here is so we took 2 groups of animals. Where first group of animals where the blood pressure is normal, and we call them Neurosciences rats, and the second group of animals where the blood pressure is high, and we call them hypertensive rats. Okay, and in both the groups, actually, we induce stroke for 1 hour, 60 minutes instead of 90 minutes. And what you can see here again, and then 24 hours later, we're doing MRI here, and what you can see in the Neurosciences group with the 60 minutes of stroke induction, there's a relatively small damage here. Whereas in the hypertensive animals, we see that even with just a small 60 minutes actually ischemic insult, there's massive damage that you can see here. And that's again represented in this bar diagram here. Now in these hypertensive animals, if we again treat them with the peptide 6 hours after the onset of the insult, we see again how substantial decrease actually in the infarct size here. And again, it is represented here in the bar diagram now. Last slide, please. So where do we stand right now in terms of our the peptide and its treatment efficacy? So we have done acute phase 3 outcome. We have measured that at 24 hours. The study is completed. We have also measured the long-term outcome, actually, which is up to 4 weeks, and that study is also completed. We have also evaluated the therapeutic time window. At this point, we can see even if we can inject the drug 6 hours after the onset of an ischemic insult, actually, it still has a significant efficacy. Finally, we are also looking at these long-term outcome studies in hypertensive conditions. We have completed the short-term work, and this work is in progress right now. So once we have this work done, our next goal is to go and generate actually GMP material. This is a peptide, so it's not a chemical. So it has to be made, and it's like TPA, it's a recombinant peptide, which is currently being done in the lab. Very time-consuming procedure. So with the GMP material, with partnership with any company, pharmaceutical company, if we can make the GMP material, the next goal will be to have the toxicology study done, and then to go for a multi-center randomized preclinical trial. And once we complete this part, I think we will be ready to go for an IND for approval to human clinical trials. Thank you so much.
Thank you, Surojit. I think we only have time for one question. I see you've done the work from between 2 and 6 hours. Do you expect that it could work after 6 hours, or do you have any data to show it would work longer?
That's a good question. We haven't tried that. So one of the things that we are trying right now is to go up to 9 hours, and then also see if we can have multiple dosage of the drug too.
Okay, great. Thank you.